Hello everyone. Hope you're all in the best of spirits, health and disposition. Welcome to a new video of Quest of Truth. Today, we are going to talk about the basic process of viral infection, which is its replicative cycle. For production of disease, viruses must recognize an appropriate target cell, enter into the cell, replicate, spread within the body, and produce cellular injury. The host immune response also plays an important role in the process of viral clearance or establishment of persistent infection and viral shedding. As we have already discussed in our previous videos, viruses do not reproduce, they multiply, and they need a living medium to multiply. This happens due to lack of biosynthetic enzymes. Viruses need to utilize the biochemical machinery of the host cell to synthesize virus-specific macromolecules, which is required for the production of virus progeny. In a way, it's hijacking the biochemical machinery of the host cell for its own benefit. The genetic information for replication is contained in the viral nucleic acid. The one-step growth curve starts with the eclipse period, which is followed by a period of exponential growth. The general outline of the replicative cycle is similar in most viruses, but the details vary from group to group. The replicative cycle of virus can be divided into 6 sequential phases, though sometimes there may be overlapping of phases. Adsorption or attachment, entry or penetration, uncoating, biosynthesis, maturation or assembly, and release. So the first stage of replicative cycle is adsorption. Viruses, like many other pathogens, are host specific. That means to say, they recognize a limited number of host cell types, allowing infection of some tissues but not others. This is also called as viral tropism. Now, the viruses come in contact with the specific host cells through random collision, but adsorption is a specific process which involves specific interactions between the molecules of nucleocapsid or the glycoprotein spikes present on the envelope of viruses and the target cell. The next sequential stage in replicative cycle is penetration. Penetration is the passage of the virus particle from the attachment site on the surface of the cell across the cell membrane and into the cytoplasm. The mechanism of penetration or internalization depends on the structure of virus and the cell type. So there are 2 general mechanisms of penetration or entry of viral cells into host cells. These are receptor-mediated endocytosis or viropexis. This is seen in most non-enveloped viruses. Next is membrane fusion. These are seen in nearly all kinds of enveloped viruses. For example, the human immunodeficiency virus or HIV virus fuse their membranes with the plasma membrane of host cell soon after attachment of the virus to the host cell. The viral nucleocapsid or genome are then released directly into the cytoplasm and the viral envelope remains inserted into the plasma membrane. In viropexis, the virus particle is engulfed by a mechanism resembling phagocytosis. The cell membrane invaginates, enclosing the virus particle in an endocytotic vesicle or endosome. Picorna and papovaviruses may enter the cell by endocytosis. The next step is uncoating, which occurs either simultaneously or shortly after penetration of the cell. Uncoating refers to the process of physical separation of viral nucleic acid from its outer structural components that enables the expression of the viral genes for carrying out replication once the virus is internalized within the host cell. Now you have guessed it right. For enveloped viruses, penetration process itself is the first step of uncoating. In non-enveloped viruses, the outer coat is removed in the phagocytic vacuole by lysosomal enzymes. The next sequential phase in replicative cycle is biosynthesis. In this phase, there occurs synthesis not merely of the viral nucleic acid and capsid protein, but also enzyme required in the various stages of viral synthesis, assembly, and release. Viruses must first synthesize mRNA for replication. The essential theme in replication of virus is that specific mRNAs must be transcribed from the viral nucleic acid. Once this is achieved, the host cell resources can be utilized for translating mRNA into components of virus particle. On the basis of the structure of their genomes, viruses use different strategies for transcription of mRNA. In double-stranded DNA viruses, for example, adenovirus, herpes virus, or papovavirus, the DNA enters the nucleus of host cell and uses the host cell enzymes for transcription. The single-stranded DNA viruses, like parvoviruses, use the host DNA polymerases for making the single-stranded DNA double-stranded. This is followed by transcription of the DNA into mRNA. In reoviruses, the double-stranded RNA is transcribed into mRNA by viral polymerases, which is enclosed in the inner capsid core. So the positive RNA transcripts are not only used for translation, but are also used as templates for complementary or negative strand synthesis, resulting in the production of double-stranded RNA progeny. The single-stranded RNA viruses are broadly classified into two categories: the positive strand or plus strand RNA viruses, or the negative strand or minus sense RNA viruses. In the positive strand RNA viruses, the viral RNA itself acts as the mRNA. Viral RNA is infectious by itself. In the early period, following entry of the virus into the cell, the viral positive RNA binds to ribosomes and is translated into viral proteins in the host cell cytoplasm. For example, Toga virus, Picorna virus, and coronaviruses. The negative strand or minus sense RNA viruses, for example, orthomyxoviruses, paramyxoviruses, or rhabdoviridae, the RNA is antisense, having polarity opposite to the mRNA. The negative strand RNA is not infectious by itself and carries its own RNA polymerase for mRNA transcription. The polymerase is associated with its genome as part of the nucleocapsid. A full-length positive strand RNA is produced by the viral RNA polymerase that acts as a template to generate more copies of the genome. Retroviridae, with a genome of single-stranded RNA of positive polarity, exhibit a unique replicative strategy. Their single-stranded RNA genome is converted into an RNA-DNA hybrid using the viral reverse transcriptase, that is RNA-directed DNA polymerase. This is reverse transcription. The double-stranded DNA is then synthesized from the RNA-DNA hybrid, which enters the nucleus and becomes integrated into the host cell chromosome by the action of a viral integrase. Viral RNA, mRNAs, and progeny positive strand RNA genomes are transcribed by the host cell RNA polymerase. The mRNA encodes early and late viral proteins. Early proteins are non-structural elements, for example enzymes, and late proteins are structural components, for example capsid, envelope. As a general rule, DNA viruses replicate and are assembled in the nucleus, while RNA viruses are assembled in the cytoplasm. Following synthesis of necessary pieces of viral proteins and viral nucleic acid, there is a stage of assembly, which is known as morphogenesis. This is followed by the release of the progeny, which is usually a passive event and results from the disintegration of the dying cell. The viruses can be transmitted through the following routes: by oral transmission, direct skin contact, transplacental, sexual transmission, etc. So, in a nutshell, the infection and replication of viruses follows the following steps: adsorption, penetration, uncoating, replication, maturation and assembly, and release.